everyone. We are going to get started. And thank you all for joining us for the July seminar on physical genomics. My name is Geng Wang here at the Center of Physical Genomics and Engineering at Northwestern University. We are very excited to welcome our invited speaker today, Professor Janet Sorrells. Yeah. And Professor Sorrells is the Donald Snyder Career Development Assistant Professor of Electrical and Systems Engineering at Wharton University in St. Louis. She received her PhD in bioengineering from the University of Illinois, Urbana-Champaign in 2024, and then started her lab at Wharton University, where she develops new techniques and applications for nonlinear optical microscopy. She has received many rewards for her work, including the 2023 GenLab Young Investigator Award, 2023 Illinois Innovation Award, 2024 Edmond Optics Ultrafast Educational Award, and most recently, 2025 NIH Director's Early Independence Award. Janet, thank you for taking the time to share your work with us today. So many awards. And a quick note for our audience, we will have a question and answer session at the conclusion of the talk. So you can type questions into the Zoom chat at any time, and we will address them during the question and answer. And if you would like to learn more about the current research and training programs here at the CPGE, please visit us at physicalgenomics.northwestern.edu. Check out our growing video archive on YouTube or follow us on Twitter, X, or Blue Sky. At this point, I will turn the Zoom over to you, Janet. Thank you again.
Thank you. All right, let me see. I can share my screen. Share this and good. Okay. Oh, now I need to hide that. Okay, great. Awesome. All right. Thank you so much for having me. I'm very excited to be here. So excited, in fact, that I know you guys normally do virtual, but I woke up at 5 a.m. to drive here from St. Louis because I hope it will be more fun that way. So yeah, great. And then also, I'm very excited to speak in this seminar series, but I don't really do physical genomics, but I think we have a lot of similar ideas and themes that are very complementary to each other. Biological systems are super complex and heterogeneous and dynamic over 3 dimensions in space and also over time. And there are a lot of different ways that we can try to investigate that. And so what my lab does is we build really cool microscopes, my opinion, just correct, in order to better examine biological systems. So my title is quite long, but my goal is that at least we can understand what every single word in this means by the end. So let's go. Oh, great. Okay. So I think most people will be familiar with fluorescence microscopy. We send in light at a given wavelength. An electron goes from the ground state to the excited state. They will lose a little bit of energy and emit light at a new wavelength. We have some problems. So if we want to image a biological system, long wavelengths travel a lot better than short ones through biological tissue. So if I do my flashlight on my phone, right, white light, all the colors. If I put my finger, only red light, longer wavelength photons are traveling through it. So if we want to image deep into a biological system, then we need to use longer wavelengths. Most of our target fluorophores will be excited with some blues or some greens. Oh no, what are we going to do? All right, more problems. we have out-of-plane background fluorescence. So we focus light, right, of a given wavelength into, let's say, a cuvette of fluorescent dye, we're going to see fluorescence from kind of the whole thing. And if we try to image this with just a camera, right, imaging 2D, and we've got a whole bunch of 3D signal, it's really hard for us to localize where that signal is coming from. We have poor spatial resolution and a really high background. Not very good. Also, we're usually, but not always, limited to image one fluorophore at a time, which could tell us about like one biomolecule within our sample. But like I said at the beginning, right, biological samples are really complicated, right? And telling us about one specific fluorophore within one sample, right, has a really limited scope in what that can tell us. So we're going to fix it. Instead of using one photon to excite fluorescence in single photon fluorescence, we can use two photons in multi-photon fluorescence, specifically two photon fluorescence. So these two photons have each half the energy, twice the wavelength of the original single photon excitation. So we solved our first problem. We're using red light or near infrared now. We're going to eliminate our out-of-plane background fluorescence because if you think about the probability of simultaneously, which by simultaneously, I mean on the femtosecond level, okay, really, really tiny, simultaneously absorbing two photons, that probability is really, really small. So it's only going to happen at a really tight focus spatially, gives us good spatial resolution. We're also going to use a pulsed laser, which is going to send a really narrow, let's say, femtosecond burst of light every 12 to 100 or even 1,000 nanoseconds. So it's very localized in time as well. And we can now not just image like a single fluorophore, which we can, but we can image multiple different things. So we can do multimodal imaging with multiple different modalities. So we can do multi-photon fluorescence, like two photon fluorescence. We can also do three. We can do something. For multi-photon fluorescence, we can excite all different types of things. This is an example of the excitation and emission profiles of NADPH and FAD. These are autofluorescent molecules. So they're inside our cells, and they're really important in metabolism. Show you a little map later. But the nice thing is that if we want to image these, they're already in all our cells or in any cell of something that is or was alive. And so all we need is our two photon excitation, and we can image them, and we can make some inferences about the metabolism of a sample. I'll explain how that works later on. We can also do harmonic generation. So this happens at certain types of materials. And one of those where it happens is in collagen. So we can image collagen, which is like really important for our extracellular matrix. Collagen remodeling happens a lot in certain types of diseases like cancer or in like liver disease. Collagen also having a resurgence for skincare. So you probably get marketed some collagen face mask or something to help your face be more firm, right? So because collagen is very important in that structure. We can also do something called coherent Raman scattering, which can tell us about the biochemical profile from Raman scattering within samples. So a quick aside on Raman scattering. So chemical bonds have a specific chemical energy or a like vibrational energy. And those are really specific and at specific frequencies. And so if we send in light of a given wavelength, right, and we can see that energy is taken or transferred to our input light. Our input light's in red, and we can either get out, right, the purple or the orange. If we're getting Stokes or anti-Stokes scattering, and the amount of energy transferred is the vibrational energy of that chemical bond. And we can do that in a hyperspectral way and collect a whole bunch of data, and then we can have really specific hyperspectral information that will tell us all about the biochemical profile of a sample. It's a lot of data. Okay. And so we can combine together multiple modalities. So this is some work from the lab where I did my PhD. And so we called this SLAM microscopy, simultaneous label-free autofluorescence multi-harmonics. That's a lot of words. And we got 4 channels. So we could look at 4 different nonlinear optical modalities. And so we can look at changes in refractive index, 3-photon autofluorescence, second harmonic generation. We got some beautiful collagen structures there, and also 2-photon excited fluorescence. And if we put these together, we can get really, really beautiful images. And these are like totally label free. So it's all just based on some inherent contrast based on the structural and the chemical profile of the sample. So this is an example from an in vivo mouse pancreatic tumor. And we compared it with that pizza slice there is the H&E histology, which is like a standard way to assess and look at biological tissues. But instead of having to do all the processing of histology, we can collect an image in minutes that can show us a whole bunch of the different features of the tissue. We can also try to quantify and assess different types of samples. So this was some work also from my PhD where we were looking at extracellular vesicles that were released in patients with breast cancer and looked at it in their urine and looking at metabolic differences in those. So we can look at quite diverse biological samples from particles in urine to like living tissues. All right. So we can call this like optical metabolic imaging, and we can look at the metabolism of samples because NADH and FAD are related to metabolism. They're super important. I'm sure many of you have taken a biology class at some point, but maybe it's been a long time. So what I remind my students, because I went from my degrees in bioengineering to joining electrical engineering department, and sometimes I'll show this and it's a little overwhelming for electrical engineers, but I just remind them that they probably remember the mitochondria as the powerhouse of the cell, and they say, oh yeah, I remember that. I'm like, okay, that's what we're imaging. That's the mitochondria. That's energy metabolism. So when I say that, it means we're going to take in glucose at the start up there, and we're going to make ATP, which means we're storing chemical energy. And in that process, we have a ton of different reactions. You don't need to memorize them all, but NADH and FAD are related, and their relative amounts and what they're doing can tell us something about metabolism. And so most of the work that's on optical metabolic imaging is imaging mammalian samples. And so we're looking at glycolysis and we're looking at the mitochondria and making inferences about metabolism. We can also look at bacteria metabolism, which is really cool. They don't have mitochondria, but they have all the same machinery, but it's just all mixed together inside the cytosol of the cell. So we can image really diverse samples, which is great. Okay, so in addition to just getting beautiful images, we probably need to quantify them. So the ways that we will generally quantify our optical metabolic imaging, the first way is we can look at this optical redox ratio. So it's like reduction oxidation ratio. And we can look at the relative intensities of our FAD and our NADH. So you can see there, I put NAD parentheses pH. A quick note to that is that NADH and NADPH, they're different molecules. The P is just a phosphate, but they do different stuff. Most of the NAD parentheses pH in our cells is NADH, so we can kind of assume that's playing more of a role, but they have basically overlapping excitation and emission and fluorescence, so we've got to group them together when we're imaging just to acknowledge that NADPH in parentheses is there. Okay. And so we can approximate that that ratio of intensities that we get is going to be proportional to the relative amount of this electron transport chain that's happening in the mitochondria activity and help us to quantify how much electron transport chain activity is happening in a given cell. Next, more fun, my favorite thing, is we can look at the fluorescence lifetime. So after we excite a molecule to the excited state, and we're waiting to see if it's going to emit a photon, there's going to be some amount of time there. And that time is going to be on the nanosecond scale. But it's not just going to be the same amount of time every time. It follows an exponential decay probability density function. So this means, right, we can look at the probability of emission, or if we image a whole bunch of times, it will mirror it. So it can be our intensity of our emission over time on the nanosecond scale. And it's going to follow this equation e to the negative t over tau, where tau is the fluorescence lifetime. And the fluorescence lifetime depends on a whole bunch of different things, and it depends on the fluorophore. Primarily for NADH, it can tell us about protein binding. So if NADH is free, unburdened, just hanging out by itself, we'll get a much shorter fluorescence lifetime, maybe something like 0.3, 0.4 nanoseconds. If it's bound to a protein, such as in the electron transport chain and it's working hard, we're going to see a much longer fluorescence lifetime, maybe 2, 3, 6 nanoseconds. And we can make a beautiful color-coded map based on the fluorescence lifetime and then map it out in different cells. So one of the things that we've used this for previously is looking at apoptosis, which is programmed cell death, in breast cancer cells. So we had a few different breast cancer cell lines and then one non-cancerous cell line on the bottom here. And we treated them with this staurosporin to induce apoptosis and looked at the dynamics that will occur, changes in intensity and lifetime, in order to see how fast that's occurring and also look at heterogeneity within the cells as it's occurring. This is also some data that I took. So this is an ex vivo mouse intestine. And so we can see there's a whole bunch going on here. And we can get various different maps, which all have a whole bunch of different information based on the intensity and the lifetime of NADPH and FAD in the intestine. And so I'm very excited, and I've started doing some more intestine imaging in my lab, and I have a grant from NIH in order to look at the metabolic heterogeneity and assess both the bacteria and the host cells within the mouse gastrointestinal environment, because both of those will exhibit some contrast. They all have autofluorescent molecules. They can all exhibit Raman scattering as well. And so we'll be able to make some inferences about the host microbe interactions in the gut. All right, so you're probably thinking now that nonlinear optical microscopy sounds perfect. And it can, we can get label-free images of any sample. They look beautiful. We can do multidimensional contrast. But as you probably suspected, there are some limitations to it. And some of the key limitations are that if we're acquiring data across different time points, so we want to do fluorescence lifetime, or we want to take hyperspectral data across different wavelengths, it's going to take a lot of time to acquire those. And that means that we're really limited in fast dynamics that we can look at. We're really limited in throughput. Now I'm a professor, it doesn't plague me anymore, but when I was a grad student, it means I have to sit in the lab in the dark for like 8 hours collecting my data. And then it's like, it's very limited as well because this is microscopy. So each field of view is really, really, really tiny, right? Microns across. Okay, so if we want to take nonlinear optical microscopy and bring it from being a really cool method that's quite inconvenient to being a really cool method that we can use to realistically investigate biological systems, look at dynamics, take high throughput images, and also help our grad students to not sit in the lab acquiring data in the dark all day, or at least have their days be more productive if they're doing that. We need to make our acquisition faster. So this has been my goal. So we can start off talking a little bit more about FLIM. So if we want to measure that fluorescence lifetime, the time in between excitation and emission in the fluorescence process, right? We're going to use a pulsed laser, remember, because we need to have that really temporally tight focus so that we're exciting with 2 photons simultaneously. So we'll send in a laser pulse. It will keep hitting the sample. Eventually, the sample may release a photon. That photon will go to our detector. An example of a detector that is most commonly used is a PMT or photomultiplier tube. And then that will go to generally a commercial card, which is for time correlated single photon counting, or TCSPC. And that card will say, oh, I got a photon count. And then it will use this time to amplitude converter, or TAC, to convert an amount of time to an amplitude, exactly like it sounds, digitize it with the ADC, and then that value goes to our computer, and then we say, okay, we got a photon, and it arrived 1 nanosecond after the laser pulse. Then we'll do that again, and we'll just keep waiting and collecting our photons one by one, discriminate them, time tag them, digitize it, and add it to our photon count histogram. Okay. Good. That's all I have patience for. So it will take a long time because we're going to build up this photon count histogram. So it's like number of photons versus time bin over a long time, over many, 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 many laser pulses. Of course, the laser pulse period, maybe it's pretty fast if it's 12.5 nanoseconds. But if we have to do this hundreds of times per pixel, and we're going to take, maybe 500 by 500 pixels, or maybe 1,000 by 1,000 pixels in our image, ooh, the time can really add up. Okay. But once we collect enough photons, we'll get this e to negative t over tau. We'll fit our tau, and we'll say, great, we got a beautiful image. All right, so why am I counting photons so slowly, right? So generally, the detection electronics, they have something that's called a dead time. So they have a limited bandwidth, which means that after one photon is counted, right, the electronics need some amount of reset time, right? They're going to send a signal to the computer. That signal has a width in time, right? If we're unlucky, that time may be 100 nanoseconds, right? Which means that for the next 100 nanoseconds, I can't count another photon. Right? And so what this means is that we need to limit the number of photons that are going to arrive at the detector so that they're spaced enough in time that we're not missing any. If we just crank up the power and we collect a whole bunch of photons in a row, what's going to happen is we're going to start missing photons, and then our decay curve is going to become distorted, and then both our intensity and our lifetime measurements are wrong. Disaster. All right. And so generally the way we'll limit it is we'll say our photon rate, which is going to be like the percent of photons per laser pulse. So 100% photon rate would mean that for every laser pulse hitting the sample, we're going to collect one photon. But our photon rate, we're going to set that to 5 to 10% is generally what the manufacturers of the TC-SPC cards recommend, which means that 90 to 95% of the time while I'm sitting in the dark collecting my data, no photon is hitting the detector. I think we have some room for improvement here. So what we can do is we can take our photon counting and time tagging electronics and not throw them in the trash, but put them somewhere else. And instead directly digitize the output of a detector and see what happens, right? Because those electronics have a limited bandwidth, so does our detector. But we can see, can we get faster data if we just do that? So we'll get something that looks kind of like this. Sure. And I looked at many of these when I was first developing my system as a graduate student. And I was like, there's some variability in the height, and I don't know what's causing that. There's also a lot of variability in like the time between signals. And so what eventually occurred to me is that these variabilities, right, means that the spacing between each of these peaks that I see tells me that I'm collecting individual photons, and each one of these peaks, right, each one of these pulses corresponds to one photon or maybe more. And the variability is actually just due to the variability of the gain of my detector. And so you can, right, just sum up all this data or block it in time and get out some measure of fluorescence intensity and lifetime, but those variabilities really, really mess it up. And so our goal for a perfect system, right, would be that if we collect one photon, hits the detector, we get a count of 1. We got 0 photons, we get a count of 0. So that was my first mission. How can I do this? So it took some investigating. How does a PMT actually work? So a photon will hit a photocathode, release an electron. That electron is then accelerated towards a dynode, which is strongly negatively charged. And that electron will hit the dynode and then boom, explode out. And we're going to get maybe 0 to 5, depending on the voltage applied to it, new electrons, which are then going to go to the next dynode, and each one multiplying until all of that charge is collected at the anode. Now, because we have this multiple dynode amplification and the gain at each dynode is quite variable, you can imagine, right? If instead of drawing those 5 initial first electrons, if I just drew 1 initial electron, we would have a lot less signal collected at the final anode, right? So our gain ends up being really, really variable, even from just 1 single photon hitting the detector. So if we turn down our power and we are sure that we're just getting single photon counts at the detector, and then we analyze what does that look like, we can now look at our gain variability and assess, right, what the peak heights are for just 1 photon at the detector. Then we can draw a little threshold and say, okay, any peak with a height above this, we're going to call photon count. There are sometimes peaks below it. Those are just from some analog noise at the bottom. And so if we look at our directly digitized data in blue, it will look like this. We add a threshold, we find a peak. A peak just means it's a pulse, and there's a value before it that's lower, and after it that's lower. That's a peak. It's above the threshold. And we're going to call that 1 photon count. So I call this method single photon peak event detection. I think it's a good enough acronym. And I liked it because I can call it speed, because it's for counting photons fast. Great. All right, so we achieved our goal. 1 photon will give us a count of 1, 0 photons, a count of 0. We got rid of our slow electronics, so now we can count photons faster. And what ends up happening is that we get data like this that's digitized, and then we can determine our photon counts. It has to go pretty fast because we're sampling maybe around 5 gigasamples per second, so that's a lot. And so I end up using GPU to accelerate my photon counting. Luckily, it's actually a pretty simple algorithm just to find a peak and a threshold. And now we get our photon counts out. Awesome. But then I got greedy, so I want to count more photons. And so then I learned about something that's called a hybrid photodetector. So it's kind of like a mix between a PMT and an APD, or avalanche photodiode. And the great thing about HPDs is they have more gain linearity and less gain variability. So if we look at our histogram of peak heights out from an HPD, where I collect the data from low to really, really high intensities, and from a PMT where I collect the data from low to really, really high intensities, we can see in our HPD data, we get these like fully discrete, but semi-discrete kind of blocks. And what we can infer from that is that this first kind of block here, all of those peaks are corresponding to like 1 single photon hitting the photocathode. And then, right, if we look at double that, we get our 2 photon peak, we can get a 3 photon, 4, and a 5. From our PMT, we don't actually have resolvable peaks. We can just say, okay, anything greater than this is greater than or equal to 1 photon hitting the detector. I tried to draw thresholds. It didn't work great. And so from our HPD data, right, we end up getting something like this. You can see there's kind of slightly more discrete levels of those peak heights from the HPD. And now we can count multiple photons at once. And so the like dead time of this system versus from traditional systems, let's say, is like 10 to 100 nanoseconds. The dead time is now limited based on our sampling frequency. So if I'm sampling at 5 gigasamples per second, I'm going to have a dead time of about 0.2 nanoseconds. And I can count multiple photons at once, so I can count photons even faster. So the system ends up looking something like this, right? It's a pretty traditional multi-photon point scanning system. The only differences from a traditional system are that our HPDs, we have 2 different spectral channels, one for NADH and one for FAD. They go to a transimpedance amplifier and then both digitized by a high-speed digitizer at 5 gigasamples per second. The con is that this is now generating data at 20 gigabytes per second. And after a couple years of fighting with my lab mates about filling up our computer really, really fast and then not transferring data off and not being able to save data, we got a high-speed digitizer with an onboard FPGA, so we could assign photon counts on the FPGA, and then use the GPU of the computer to compress the data even more so we could collect data in real time and save much more reasonably sized files of our fast FLIM. And so we're able to get 2 channels, and then I have to include this one because with Gang, who's here in the audience, we developed one system that could actually do 3 channels, and we did 2 photon excited FLIM of FAD, 3 photon of NADH, and 4 photon FLIM of tryptophan. So this was a very impressive feat that took us a long time to get. All right, so kind of to summarize this section, so we can use this speed method, right? We're bypassing these traditional slow analog electronics and using instead a high-speed digitizer and an HPD. We're performing the photon counting directly on that digitized data in the digital domain. And the kind of limit that we could work up to is about a 223% photon rate, which, if you remember back from my initial FLIM slide, most systems are collecting about a 5 to 10% photon rate, and above that, we're going to get inaccurate results. So this is a huge improvement over what traditional systems can do, and we can look at fast dynamics of apoptosis in some cancer cells, which is on the which side? Left side, and then some C. elegans worms that are swimming around on the right side. And this was one cover that we had for this work. Okay, great. So it works really well, amazing. We can collect data super, super fast. But by we, I mean me, because I can build and calibrate the system. And, of course, I dream that we can get more of these systems out, and maybe it could be commercialized one day. Someone else can use it. And I've talked to other people about helping to build and create one. But at least for right now, I think not everyone is ready to adopt the speed method just yet. So if we want to enable everyone to be able to do FLIM faster, what can we do? So the main limiting factor is the dead time of these systems, right? And every FLIM system will have a dead time, and it will just come on the spec sheet. It'll tell you what the dead time is. So is it possible that if we know our dead time, right, we know that we're going to lose photons, can we just correct for that in post-processing instead of building a custom system, which I think is more fun, but a much more reasonable approach will be, can we correct it? So Yidan, who's a PhD student in my lab, developed this method for dead time correction. And so it's based on this work from Coates in like the 1970s, but because our FLIM systems have advanced much since then, we had to do a few improvements on it, but now it's working very well. So our goal is that we want to, instead of looking at the number of photons in each time bin, so each time pulse here, we can think about the probability of detection. So we know if I collect a whole bunch of photons in, let's say, my first time bin, each time I collect a photon in the first time bin, that means the system was dead for example, 10 nanoseconds afterwards. So if I collect a photon in my first time bin, I know that my system was dead in the second time bin. So instead of, let's say in my first time bin, I collected 100 photons out of 1,000 laser pulses. My second time bin, I collected 5 photons. Instead of saying I collected 5 photons out of 1,000 laser pulses, the system was dead for 100 of those. I can collect 5 photons out of 900 laser pulses and correct it based on that. So we're trying to get our corrected photon emission probability. So we'll take the number of photons that we counted at a given time bin, and we'll just put it over the number of excitation cycles where we were not dead. And again, so we're going to take the total number of cycles and then subtract the total number of times we were dead. And we know exactly how many times the system was dead because we know the dead time. So what we can do is for each target time bin, so that's where the red arrow is on the diagram, we can then look at the 10 nanoseconds before it. And so we do a little like circ shift of our data, and then we sum all of that data in yellow, and we say the number of photons collected in the preceding 10 nanoseconds is the number of times the system was dead. And then we can correct the probability based on that. So it ends up working quite well. So there's a lot on this plot here, and it's actually quite difficult because the corrections work slightly differently for every different intensity and every different lifetime and every different dead time. So I'm just going to show you a couple examples of them for now. So our blue is like our ground truth, no dead time. Our orange is going to be with dead time, so that's uncorrected. And so this is simulating a system with a dead time of 5 nanoseconds, which is actually a pretty good system. On this plot here, right, we have our measured intensity, and on the x-axis, we have our photon rate, which is going to be our number of photons we collected. And so what happens is that as we get brighter and brighter and brighter, we're going to lose more and more photons in our uncorrected data. If we use the original Coates method, which I didn't show you the equation for, it kind of works, but it falls off. And then for Yidon's method that we developed in my lab, we call this the cross-cycle method. In green, you can see we're really, really close to that no dead time. And we decided we'll put a plus minus 10% is our acceptable error. And so we can correct data up to 600% photon rate using this method. And we can look at what the decay curves look like. The corrected decay curve, you can see on the bottom here in green. It's a little hard to see the colors, but I'll tell you, it follows the correct shape, but it's a bit noisy. And that's because, right, we're calculating that decay curve from our with dead time data, which has a lot fewer photon counts. So it does end up being a little more noisy, but overall it works. And we can get the correct intensity and also the correct lifetime out. And this is just a slightly different case with the 10 nanosecond lifetime. You can see again, it's more noisy, but we're still getting the correct lifetime. So we've collected a little bit, but not quite enough experimental data to validate Yidon's simulation. So it's work in progress, but very exciting. So with our dead time correction, this can work on any film system with a known dead time. So it's pretty universally applicable. In some of our simulations and in a few of our experimental data, we're able to get the system to work like 10 times better. So if it previously fell off at a 50% photon rate, now we can get accurate performance up to 500% photon rate, which is huge, right? This is a huge improvement. Will let us image, let's say, 10 times faster, or let us image a much larger dynamic range. And because it's post-processing, it's completely free. It doesn't require any extra calibration or other steps like the speed method does. And it's gonna, save money and is pretty easy to implement. Okay. All right. Oh, one thing I added because I wanted to speak to especially this crowd about it, is the role of NADPH in the nucleus of cells. So especially in optical metabolic imaging, when we're doing NADH and FAD autofluorescence, all of our cell diagrams look like this. You guys probably noticed there's no nucleus in this cell that I drew, right? But in our cell images, of course, so that red spot in the middle is the nucleus. And we can see, okay, there's some NADPH in there. If we look at some literature, we can see that NADH and NADPH can regulate gene expression in the nucleus. So we know that they could be doing something, right? They have specific roles relating to sirtuins, right, which can play a role in aging, transcription, chromatin structure. Any chromatin structure enthusiasts in the audience? Okay. But optical imaging of NADH and NADPH is like in the nucleus is super underexplored. So what a lot of groups will do is they'll take cell images like my beautiful image here, and then they'll just mask out the nucleus, and then they'll do all their analysis to talk about the metabolism and the cytosol and the mitochondria, and then just ignore anything relating to the nucleus. So I think there's more that can be done. But I think it's still an open question. So we should talk more about it. All right. So I'll move on a little bit to talk a little more about Raman scattering. So this is the slide I already showed, right? We can get information based on the vibrational energy of different chemical bonds, but spontaneous Raman scattering is generally really, really slow. Too slow that we can't, or if any of you guys have used the confocal Raman, okay, did you have to like wait overnight for it to collect one image? I'm not very patient. So we can do something called coherent Raman scattering by using nonlinear optics. So we're going to send in two laser pulses, a pump beam and a Stokes beam. Okay, and one thing is called Cars, but it's not the Pixar movie. So we'll send in two beams, pump beam and the Stokes beam of our pulse laser, and we're going to be able to get out our anti-Stokes photon based on the vibrational frequency difference between our pump and our Stokes beam, and then we can collect hyperspectral data and get images. And this is going to happen depending about 100 to 10,000 times faster than spontaneous Raman. So this is great. But it can still be a little slow. So what we're going to do is we want to look at the instantaneous frequency difference. So these plots is showing the frequency. So my two beams, I gave them wavelengths, but they both have a little bit of width in frequency and a little bit of width in time. The instantaneous frequency difference, so if we look at, right, the length of that arrow where it says omega 1, right, this is going to be the frequency that we're examining in our sample. And normally we'll have a specific delay between the two beams. We'll take an image, change the delay, take an image, change the delay, take an image, right? And we can build up, right, a hyperspectral data cube across each different wavelength. This again will be pretty tiring. I didn't even have the patience to put more than like 10 spots on this. But normally we were interested in like hundreds or thousands of wave numbers, not just 10. So is there a way that we can do this more efficiently? Probably you guys guessed, the answer is yes. So we can do something called ultra-fast pulse shaping. And so what we're going to do is we're going to take a beam in, separate it out spatially by wavelength with a grating, send all those wavelengths to a spatial light modulator where we can then adjust the phase delay of each of those wavelengths independently. And now we can control the phase, so move it back or forth in time, right? And what we can also do is apply certain patterns that are going to adjust the amplitude. So if there's a wavelength that we don't want, we can silence that. We can create destructive interference in the far field, and we're going to cut out that wavelength that we don't want. Now we have perfect control over our excitation pulses, and we can control them in anywhere within this frequency time space to get whatever excitation we want. So our first implementation we tested was just to see, can we get some spectral scanning? And so instead of tuning the time delay between two pulses, we instead, right, within our I call this our like window of possibility, right? The light purple tells us we can have excitation anywhere kind of within that oval. And the purple indicates the excitation that we've chosen, right? And so we can scan our excitation frequency by choosing different ones within that and look at different wave numbers or different frequencies. Okay, so it worked, but that's still one by one collection. So we need to have a little more fun with it. So instead of doing some spectral sweeping, what we're going to do now is spectrally tailored excitation. So instead of just choosing one frequency band within our window of possibility, we can create a hyperspectral pattern. And we can choose whatever we want and apply that pattern onto the SLM and then get out whatever we want in the frequency space. And so this allows us to choose patterns that will go up and down in frequency. We can have broader, we can have narrower, we can do whatever we want, total freedom. And so this is creating a hyperspectral excitation envelope. So it's not just one wave number, right? It's some pattern in intensity versus frequency, and we can choose whatever we want. Okay. When I first started with this idea, I got some pushback saying, why would you do this? Is it reasonable, and can we generate hyperspectral contrast if we just get a few broad channels, right? If you get a few different excitation patterns that are custom, how could that tell us more information than having a whole bunch of single frequency data points? The thing is that my inspiration for this is the human eye, right? So most of us have 3 different spectral channels in our eyes, right? And those are designed by evolution in order to let us see a really broad range of colors. So can we come up with some set of hyperspectral excitation envelopes, such like our cones have been, in order to generate hyperspectral contrast, but just using many fewer spectral channels that are smartly designed. So it turns out we can. And so this is just an example showing, okay, our slow hyperspectral sweeping going one by one, so it takes so long, versus our spectrally tailored patterns, we can get this hyperspectral excitation envelope in whatever shape we like. First, we do some proof of concept. I got 2 different types of beads. I measured each of their spectra alone, and then I mixed them together. And I created 2 masks. Those are the solid lines in orange and blue. And then applied those masks. I took 2 images and then made a little scatter plot and segmented out the beads. And then I also took a hyperspectral image and segmented it out to validate, and it matched very well. So instead of having to take 100 different wave numbers to separate out my 2 different types of beads, with prior knowledge of the beads that I was going to be looking for, I was able to separate out the beads from each other with just 2 images instead. So we made it 50 times more efficient. I also did some similar images looking at some adipocytes, and so just from like the underside of some ex vivo mouse skin, and tried different pairs of excitation masks, made a little pixel-wise scatter plot, and segmented out different regions within those cells. And I think this graphic is actually a little better. It just shows different excitation masks that I chose, and then what the images that correspond to those. So it's like each one is a single shot. I create a mask, I send it to the SLM, and then we take one image with that mask, and that's what the images look like. So the key here is that we have flexible, untunable acquisition. We can program it for whatever type of excitation we want. For the previous examples that I showed, right, I had prior knowledge of what I was imaging, but we don't always have that. And so another thing I made is a few different sets that I can image with. So one is a high resolution sweep. So this is going to go through 100 different wave numbers and collect our best spectral resolution image. And so we want this if we need to know really precisely, like, what's the biochemical profile of this sample. Then I made a lower resolution sweep set that just had 10 images. So we can image 10 times faster, but we get worse spectral resolution on purpose, because for certain imaging tasks, right, we have to complete that in a certain amount of time. Maybe the dynamics are happening fast, maybe the sample's going to die, maybe we need to image 100 samples before the end of the day, and there's only a few hours left in the day, right? I also, for fun, made some Fourier component patterns, which are different sines and cosines at different frequencies. They ended up with some big reconstruction artifacts. But if we look at it on kind of this map of the signal magnitude, spectral resolution, and the acquisition time, we can see that we've got the ability to play and mess around within this space. And so we can customize based on our requirements for what we want to image as well. So this is without any prior knowledge of what we're imaging, but knowing our constraints on spectral resolution, SNR, and time. So this gives us like a control over our hyperspectral excitation envelope. Within this whole space, we can tailor it for specific things, or we can tailor it for specific spectra if we know something about our sample beforehand, which actually we often do because the Raman spectra, you can just Google what's the Raman spectra of a certain thing if you want to check it, or many smart chemistry people, they already know it. Or we can just tune it based on, okay, I have this much time left in my day. This is how many samples that I want to image. Here's the spectral resolution I can manage. So we can generally achieve higher throughput imaging with this, and I think it's got broad applications throughout all of hyperspectral imaging. There's like 100 slides of ultra-fast optics that I've skipped here. not just in the presentation, but also in the first implementation of the work. So one of my students, Trufan, is working through that, and there's a whole bunch of extra physics that we're now considering in order to make things a lot more accurate. Okay, so kind of to summarize, we can accelerate the temporal and spectral aspects of our acquisition in order to expand our capabilities. We can use the speed method and computational photon counting to still have single photon precision, but image much, much, much faster. We can do dead time correction on any system, which also allows us to image much faster. So we can look at fast dynamics and do higher throughput imaging without sacrificing our accuracy. And for hyperspectral imaging, we can control the phase and amplitude of our excitation light and control these hyperspectral excitation envelopes however we want. So just to summarize, like kind of the key themes that we're going on is that we have all this high dimensional data that traditionally we collect, but usually in post, we'll compress that into its useful components, right? Not only in time and in spectra, but also sometimes in space. And so the key goals of my lab is developing ways that we can incorporate that data compression into our acquisition system in order to collect data more efficiently, save time on acquisition and processing, and make everything more convenient and nicer. All right. That's it. I think I talked a little too fast at the end, but I made it in time. So I'll take any questions now, and if anything occurs to you later on, feel free to email me as well. Oh, and please let me thank my graduate students. They're all so wonderful. And also Steve Bopard, who was my PhD advisor, and really enabled all of this work and has also been a great mentor as I'm starting my own lab.
Yeah, great talk.
For the people on Zoom, if you have questions, just feel free to unmute yourself and ask or type in the chat. Or for the audience here, it'll be nice if you can just come here just so that we get the audio on the Zoom for posting. So if you have questions, just...
Or if you're shy, I can repeat the question, but please come up if you would like.
Thank you for your talk. It was super engaging and very clear. Thank you for sharing all that. So I'm Ravana, I'm a grad student in the Bachman and Schleifer groups. And one of the projects we think about is ion dysregulation and disease, and thinking about how we can target the ion concentration to remodel chromatin to help to cure it. So one question I just have is for your NADH studies, do you see ion differences or dysregulation affect the fluorescence lifetime?
That's a great question. So the I'll give you two incomplete answers. So the first thing is that fluorescence lifetime is sensitive to like any nanoscale factor, right, including ion concentration. So theoretically, yes, fluorescence lifetime is sensitive to ion concentration. Do I see ion concentration dependent differences? I don't know. I see differences in fluorescence lifetime all the time. Sometimes I know what caused them, sometimes I don't. So I think fluorescence lifetime could be a great thing to look at. I think there would need to be some special control experiments to make sure that that's what it is. And there's also different like ion concentration dependent FLIM sensors that could be better for like specifics. But yeah, that could be something that's causing a difference in the nuclear fluorescence lifetime of NADH, and somebody's got to look into that. Other questions? Yeah.
It's linked to can you actually ion concentration?
So the question was, can we determine ion concentration in the nucleus? So like, could I do that tomorrow? No. Do I think it's possible? Probably. I'm thinking one of my collaborators who's in neuroscience, so she has like a calcium sensor where the fluorescence lifetime will change based on the influx or efflux of these calcium ions in neurons. And I'm trying to think if she's able to calculate the concentration or if it's still relative, but I'm not sure. But that's a good question. Someone should look into that. Yes. So, I mean, it's quite possible to calibrate like certain things, but then because fluorescence lifetime is sensitive to multiple things, then it becomes a little sticky, right? So if the temperature is different, which actually the temperature in our mitochondria is different than the rest of the cell, right? Then the fluorescence lifetime will be different as well. And so if it could be hard to decouple precisely whether it's like a protein binding or an ion concentration difference from NADH. But it could be possible. We just need a lot of calibration and maybe some validation with a separate ion sensor. Huh? I think we could do it though. How hard could it be? Anyways, actually I'm joking. It will probably be really hard and take years of work, but maybe it's possible. We can talk more. Other questions? Oh, I can't see the chat on my computer. Yes.
Beautiful. But abstract same things on that, there be clinical samples. The samples need for molecular generate clinical variety.
Okay. So the question was if these methods are compatible with like clinical FFPE samples. Yeah. So, yes and no. So, right, generally, depending on what type of processing, whether it's fixation, paraffin embedding, flash freezing, or just an ex vivo sample that's been sitting out on the counter for however long while I'm taking my lunch break, something is going to change between when a tissue is taken out of a body where it's in tight homeostasis, and then it's no longer that way. And because our fluorescence lifetime is dependent on so many different factors, right? It will be different. However, as long as there's NADH and FAD in that sample, if they don't get washed out, it's possible to image. And there's one study by Irene Georgiakoudi's group, formerly at Tufts, now at Dartmouth. Okay, I forgot the last name of the first author, but her first name is Adriana. And she compared actually, I think it was fresh ex vivo samples and then the FFPE samples to look at. And there are differences in the fluorescence intensity and lifetime, but I believe it can be at least somewhat accounted for and calibrated for. In terms of cars, I believe it should be possible. It also depends on what type of, if there's going to be any extra background, because pretty much everything has chemical bonds and can produce some Raman signal. And so it will just be whether or not the signal to background is good enough. But I think it should be. One of the key benefits is that these methods can be done on live samples. And so like the translation from directly live to anything that's had something done to it or is dead is not one-to-one, but they can still work on any process sample as well. Good question. Other questions? Oh, one more.
Yeah. I want to know, in your brain here, if it could be in, do you see like a future in it? You talked about virtualizing products, maybe, yes, across more university labs or academic spaces, but what about in clinical space? What would you envision?
Oh, wonderful. Okay, so the question was about like clinical translation of this work. Yes. So yeah, there's a few barriers, right? Systems are hard to build, they're expensive. A lot of the laser sources, like the cheapest femtosecond lasers, will be maybe $60,000, $80,000, and the nicest ones are like close to $400,000. So that's one barrier, right? It's never going to be in every pediatrician's office. Well, maybe we'll see, but the laser people have to do a lot more work before we could think about that. But in terms of clinical translation, I'm really proud of my former advisor and his group right now, Steve Bopart. They have one of the ARPA-H grants for like $30 million, and they're developing a clinical system for tumor margin assessment using optical coherence microscopy and nonlinear optical microscopy. So developing these methods to be able to rapidly assess tumor margins like intraoperatively. And so because it has high resolution, it doesn't require any staining, and it can be done on live tissues. It lends itself well to that type of problem, but it's really difficult. So for like my lab starting out, I'm not ready for any huge clinical translational studies like that yet, but they've done amazing progress in just the past couple of years in developing this clinical system. So maybe, hopefully it will work. Great question. Awesome. If that's it, thank you guys so much.
Thank you.